Can you see my screen?
Yeah, ma'am.
All right. Okay, so cell, everybody knows that cell is the basic unit of life, isn't it? If now on the basis of the cells, there are two types of animals or organisms in general. Do you know what are the two types of organisms? Okay, so on the basis of the number of cells present in the organisms, there are two types of organisms. One is unicellular organisms, and then there is multicellular organisms. So unicellular organisms are those organisms that just have a single cell in their entire body, whereas the multicellular organisms are those that have many cells in their body. Okay. Now, what happens? Sometimes these cells, they come up together and they form a tissue, and then these tissues, they group together, then form an organ. And when several organs group together, they form an organ system. Is it clear to everybody? Hello, is it clear, guys?
Yes, ma'am.
All right. Okay, so let us see what the NCERT says. So what is a cell? So a unicellular organism is capable of independent existence and performing the essential functions of life. Anything less than a complete structure of a cell does not ensure independent living, meaning any organism that does not have a cell, that organism is not counted as living. Rather, it is counted as non-living. But any organism that even has, one cell in their body, and then they are, completely independently existing, meaning they are present alone with the help of that cell. So this is what, the basic structure means, meaning if even one cell is there in the organism's body, then yes, it can ensure the independent existence. Okay. Now, who discovered a living cell? So Anton von Leeuwenhoek, he was the first person who saw and discovered a live cell, and Robert Brown later on discovered the nucleus. So Robert Brown is known for his discovery of the nucleus. The invention of the microscope and its improvement leading to the electron microscope revealed all the structure details of the cell. Now see, earlier in those times, there were, very much less technologies. And with respect to microscope, there was, not much magnification even there in the microscope. So a lot of things about the cells we were not able to see. So after the improvement of the microscope to the electron microscope, which is highly precise, then we can find out different structural details of the cell, like whatever things are there in the cell, then we can find it out. Then talking about the cell theory. So in the year 1838, Matthias Kleiden, who was a German botanist, examined a large number of plants and observed that all plants are composed of different kinds of cells which form the tissues of the plant. Okay, so meaning every single plant that is there, they all are composed of, different kinds of cells, and those cells, when they come together, they form the tissues of the plant. Okay, now at the same time, there was a person whose name was Theodore Spawn in the year 1839. He was a British zoologist. He studied different types of animal cells and reported that the cells, they had a thin outer layer, which is today known as the plasma membrane. So he was the primarily, the person who discovered the plasma membrane, at that time. Okay. Now, he also concluded, based on his studies on the plant tissue, that the presence of cell wall is a unique character of the plant cells. Okay, cell wall is present only in the plant cells and not in the animal cells. So on the basis of all these points that these two scientists, or botanists and zoologists stated, they proposed a cell theory based off of this. Okay, so Kleiden and Swan together formulated the cell theory. This theory, however, did not explain as to how new cells were formed. Right? They did tell that what kind, like how many cells are there. They even said that, plasma membrane is there, then cell wall is there, but they did not tell us that how those new cells were being formed. So then came another scientist whose name was Rudolf Virchow, and in the year 1855, he explained that the cells that are newly formed, they came from the pre-existing cells, meaning the cells that have been existing already, they gave rise to the new cells. And this phenomena, wherein the new cells are formed from the pre-existing cell, it is known as the ominous cellular e-cellular. Okay. So finally, this gave, a final shape to the cell theory. And finally, the proponents, or the three, like the points of the cell theory, are understood today as all the living organisms are composed of cells and product of cells, and then all the cells arise from pre-existing cells. Okay, these are the points of the cell theory. Is it clear? Hello? Am I audible? Hello? Students, am I audible to you?
Yes, ma'am. Yeah, yeah, yeah.
Okay, at least respond so that I'll know you people are here. Okay. All right, so let us see an overview of the cell according to this particular book. So you have earlier observed cells in an onion peel or a human cheek cells. Now see, whenever we, see the cells under a microscope, we can find out the structure, the size, the shape of the cell. Okay, so when we saw the onion peel under the microscope, we found that they were made of, all the parts of that particular piece or the onion peel was made up of same types of cells. And when we found, like when we saw the human cheek cells under the microscope, then we found that there was a dense membrane bound nucleus there. Okay, and also in the animal cell, obviously there is no cell wall present, whereas in the plant cell wall will be there. Okay, now the nucleus, which is a very darkly stained structure. See, whenever we try to see anything under a microscope, okay, any cell under a microscope, since all of those are sort of transparent, and obviously we cannot see it with our naked eye. So what we do before reading it or studying it under microscope, what do we do? Can anybody give me the answer?
Okay, repeat. Please.
Yes, I am asking, like for example, you want to study a cell, a particular cell under a microscope. So what you will do so that that particular cell will be visually, visible to you? Siddhika, do you know the answer?
No, ma'am.
Okay, see, what happens whenever we, see any kind of cell under a microscope, the first thing to know that, not all of the cells are clearly visible as such. Many cells are transparent. So what we do, we stain the cells, okay? Meaning we color the cells with some dyes so that, it is clearly visible to us, okay? Yes, Alia, you're right. We stain it. Yes, we stain it. Now see, whenever we stain the cells, the part which contains, the maximum, which retains the maximum amount of stain is the nucleus. Okay, it is a very darkly stained area. Okay, and so inside each cell, a dense membrane bound structure is there, which is known as the nucleus. Okay, and as you know, that nucleus is the sort of, the whole control house of the cell. It controls all the activities of the cell. Okay, and inside the nucleus, chromosomes are present. Okay, and chromosomes are the carrier of the genetic material, and the genetic material is DNA. Okay, meaning cell is there, then inside the cell nucleus is present, inside the nucleus chromosomes are present, and then inside the chromosomes, or the chromosomes carry the genetic material, which is the DNA. Okay, now cells that have a membrane bound nucleus, okay, meaning a nucleus that contains a membrane surrounding it, those types of cells are known as the eukaryotic cell. Whereas those cells that do not have a membrane bound nucleus, okay, that do not have, those are known as the prokaryotic cell. Is it clear? Now, in both of the cells, whether we are talking about the prokaryotic cell or the eukaryotic cell, there is a semi-fluid matrix there, which is basically the arena of all the cellular activities, and that particular part is known as the cytoplasm of the cell. Okay, and it is there in the plants also, it is there in the animal cells also, and in the prokaryotes and eukaryotes both. Okay, now various chemical reactions occur in it to keep the cell in the living state. So see, mainly all of the things that takes place in the cell, they basically occur in the cytoplasmic area only. Why? Because cytoplasm is the main arena of all sorts of cellular activities, and this is true for both plants and animals. Is it clear to everybody?
Yes, ma'am.
Okay. Now, besides the nucleus, okay, the eukaryotic cells, obviously they have membrane bound structures that are known as organelles. Okay, organelles are also there in the cells. And what are those organelles? So the organelles are endoplasmic reticulum, there is the Golgi complex, lysosomes, mitochondria, microbodies, vacuoles. All of these structures are there in the eukaryotic cells. But when we are talking about the prokaryotic cells, so the prokaryotic cells lack the membrane bound organelles. You know, apart from a membrane around the nucleus, the prokaryotic cells also lack the membrane bound organelles. Okay, now let us talk about the ribosomes. Okay, now see, ribosomes, these are the non-membrane bound organelles found in all cells. It means that the ribosomes are found in the eukaryotic cell also and the prokaryotic cell also. Why? Because it is a non-membrane bound organelle. Okay, now within the cell, the ribosomes are found not only in the cytoplasm but also with two organelles, meaning the ribosome is not only present freely in the the cell, but apart from that it is present on the other organelles also, and those organelles are chloroplast in the case of plants and mitochondria in the case of animals, and also on the rough endoplasmic reticulum. Obviously this is the most famous spot, isn't it? So can anybody tell me, is the chloroplast present in any animal? Is there any animal that contains the chloroplast?
No.
No, that's right. No animal contains, because it is a plant organelle. Okay. Now, animal cells contain another non-membrane bound organelle, which is known as centriole. Okay, so you must remember that centriole and ribosomes are those two non-membrane bound organelles that are found in animals. And amongst them, the centriole is found only in the animals. Okay, and they are not found in, like, basically the centrioles are found in the animal cells. And what is the function of centrioles? So they help in the process of cell division. Okay, now since there are different kinds of animals, different kinds of organisms present on the earth, so the size of those organisms varies a lot, isn't it? There are some organisms that are so tiny, then there are some organisms that are really small. So talking about their cells, so the cells also would differ in size, it would differ in activities, and the form and the shape it carries, isn't it? So the cells differ greatly in size, shape, and activities. For example, the mycoplasma. Everybody knows about the mycoplasma. They have the smallest cells, and their cells, the length of their cell is only 0.3 micrometer in length. Okay. While talking about the bacteria could be 3 to 5 micrometers. So just imagine how small a mycoplasma cell would be, isn't it? These are the smallest cells, whereas even bacteria are bigger than them. Okay. So the bacterial cell could be 3 to 5 micrometers. And this was about the small. And then the largest single isolated cell is the egg of an ostrich. Okay. Now, among multicellular organisms, human red blood cells are about 7 micrometer in diameter. Nerve cells are some of the longest cells. Okay, nerve cells are really long. Now, cells vary greatly in their shape. They may be disc-like, they may be polygonal, columnar, cuboid, thread-like, or even irregular shaped cells are also there. Okay, and this shape of the cell, it varies according to the function they perform. Meaning, for example, the RBCs of the human, meaning the human red blood cells, they are biconcave. Okay, they are biconcave. So there is a reason why they are biconcave. Okay, so the shape of the cell, it varies according to the function they perform. Is it clear to everybody?
Yes,
Yes,
ma'am.
sir.
Okay, very good. Now, moving forward, let us talk about the prokaryotic cells. So the prokaryotic cells, they are represented by the bacteria, the blue-green algae, the mycoplasma, and the pleuropneumonia-like organism. Okay, so these are basically these organisms. They come under the kingdom Monera, if you remember. So these are representing the prokaryotic cells. Now, they are generally smaller and they multiply more rapidly than eukaryotic cells. Okay, let us talk about how the prokaryotic cells divide. So first of all, the size of the prokaryotic cells are very small, and then they divide more rapidly as compared to the eukaryotes. Okay, and their shape and size vary a lot. Okay, and talking about the four like shapes of the bacteria, so the bacteria are either bacillus meaning broad shaped, coccus meaning spherical, vibrio means comma shaped, and then there is spirillum, which means a spirally shaped bacteria. Okay, now since prokaryotic cells are simpler as compared to eukaryotic cell, so the organization of the prokaryotic cell is very very simpler too. Okay, and they exhibit a wide variety of shapes and sizes. Okay, shapes and sizes and functions also. As I told you that the variety of shapes are there because of the difference in the functions they perform. Okay, now the first point to note down here is that all prokaryotic cells, they have a cell wall surrounding the cell membrane, except in the case of mycoplasma. So meaning the bacteria, the blue-green algae, then there is the PPLO, all of those will have a cell wall surrounding them. But if you're talking about the mycoplasma, then no, they won't have a cell wall. Now, the fluid matrix here is also known as the cytoplasm in the prokaryotic cell. Then obviously the defining feature of the prokaryote, it's the there is no well-defined nucleus there. Meaning, what is the meaning of well-defined nucleus? Can anybody explain?
Sorry, could you repeat?
Yes, I'm saying what is the meaning of a well-defined nucleus? Tell me. See, a well-defined nucleus is the one that has a membrane surrounding it, meaning a membrane which is separating it from the rest of the cell. Okay, that is a well-defined nucleus, but the prokaryotes do not have it. Okay, so if the membrane is not present around the nucleus, it means that whatever is the content, like the content of the nucleus, is not protected by a membrane, and it is basically naked. Naked meaning it is light like that in the cell without any cover. So basically, what is the content of the nucleus? Which is the genetic material, and it is basically naked, and it is not enveloped by a nuclear membrane. Okay, so if anything does not have a cover on it, so isn't that thing exposed? So same thing happens with the content of the nucleus also. The content of the nucleus is basically the genetic material, and the genetic material in the case of prokaryotic cells are basically naked. Okay, so that is why they are lying freely in the cell. Now, in addition to the genomic DNA, which is the single chromosome or the circular DNA, many bacteria have small circular DNA outside the genomic DNA. Okay, and those are known as plasmids. Okay, so these smaller DNA, they are known as plasmids. So the bacteria, the prokaryotic cells, they have an extra DNA apart from the genomic DNA, and that DNA is known as the plasmid. Okay, now what is the function? So the plasmid DNA, it offers many unique phenotypic characters. Okay, it offers many different types of characters to the prokaryotic cell. Okay, and more specifically to the bacteria. And what is that phenotypic character? So the one such character is resistance to antibiotics. Okay, it means that no antibiotic can kill it. Okay, now DNA, this plasmid DNA is also used to monitor bacterial transformation with foreign DNA. But this is something you will learn in the higher class. Now, nuclear membrane is found in eukaryotes. No organelles like the one in eukaryotes are found in prokaryotic cells except for the ribosome. Okay. Prokaryotes have something unique in the form of inclusion. Okay. Inclusion bodies are there in the case of prokaryotes. Then a specialized differentiated form of a cell membrane called as the mesosome is the characteristic feature of prokaryote. Okay, and they are essentially infoldings of the cell membrane. See, mesosomes are basically the invagination of the cell membrane. For example, there is a cell, for example, this is the cell wall. Okay, and this is the cell membrane that I'm drawing. Then the invagination of the cell membrane or the infolding of the cell membrane, this thing will be called as the mesosome. Is it clear to everybody? So this was all about your prokaryotic cell. Okay. Then talking about the cell envelope and its modifications. Now, in most of the prokaryotic cells, particularly the bacterial cells, they have a chemically complex cell envelope. Chemically complex cell envelope. Now, the cell envelope consists of a tightly bound three-layered structure. So this cell envelope is three-layered. So the outermost layer is formed by the glycocalyx, and then it is followed by the cell wall, and finally the innermost layer is the plasma membrane. So the three layers would be glycocalyx, it would be cell wall, the second layer, and then the third layer would be the plasma membrane. Now, although each layer of the envelope forms a distinct function, okay, they would, like every layer which is present in the cell envelope, it has a different function to perform. Okay, but when they are all together, what is their function? They act as a single protective unit. Okay, now bacteria can be classified into two groups on the basis of the differences in the cell envelope and how they respond to a stain. Whenever we stain a bacteria, so we stain it with the Gram stain. Okay, Gram staining is the technique through which we can stain the bacteria. Okay, this is the staining technique of the bacteria. So those bacteria that take up the Gram stain, meaning that get stained, those are known as the Gram positive bacteria, and the others that do not take up the stain, those are known as the gram-negative bacteria. Now, talking about the glycocalyx. So the glycocalyx differs in composition and thickness among different bacteria. So in different bacteria, it could be in different thickness and different composition. So it could be a loose sheath. When the glycocalyx is present as a loose sheath, it is known as the slime layer. Okay. While when it is thick and tough, it is known as the capsule. So when it is a loose sheath, it is called as slime layer. While when it is, thick and tough, it is known as the capsule. So the cell wall determines the shape of the cell. First of all, like we are seeing, what is the function of cell wall now, which is the second layer of the cell envelope. Okay, so the cell wall determines the shape of the cell, and it provides a strong structural support to prevent the bacterium from bursting or collapsing. First of all, what does the cell wall do? It provides a definite shape to the cell. Also, it helps or protects the bacteria from bursting or from shrinking or collapsing. Okay. Now talking about the third layer, which is the plasma membrane. So plasma membrane is the semi-permeable membrane. Okay, the nature is semi-permeable and it interacts with the outside world. Now this membrane is similar structure to that of eukaryotes. Even eukaryotes have a plasma membrane that is semi-permeable in nature. Now, talking about a special membranous structure, which is known as the mesosome. Now, this is formed by the extensions of the plasma membrane into the cell. These extensions are in the form of vesicles, tubules, or lamellae. Okay. And what is the function? They help in the cell wall formation. We are talking about the functions of mesosome. So, first of all, they help in the cell wall formation. Then after the formation of cell wall, they help in DNA replication and distribution of that replicated DNA to the daughter cells, which are the offspring. Okay. Now they also help in the process of respiration, secretion, also to increase the surface area. Now anything that is folded, that increases the surface area as such. Okay. So the surface area of the plasma membrane is also increased by that. Also the enzymatic content. Now, in some prokaryotes like cyanobacteria, which are also known as the blue-green algae, there are other membranous extensions into the cytoplasm. And the membranous extensions of the cyanobacteria or the blue-green algae, they are known as the chromatophores. Okay. Now, anything where there is chrome related. So it means pigments. So chromatophores, those membranous extensions that contain the pigments are known as chromatophores. And same, this membrane extension without pigment would be mesosome. Okay. Now, bacterial cell can be motile or non-motile, meaning if motile, they have a filament extension from their cell wall, which is known as a flagella. Okay, with the help of flagella, the bacteria can move. Okay. Now, the bacteria show a range in the number and arrangement of the flagella. Bacterial flagellum is composed of three parts. The three parts of the filaments, the three parts of the flagellum are filament, hook, and basal body. The filament is the longest portion and extends from the cell to the cell surface to the outside. Okay, the longest portion is the filament. Now this was about the flagella and what is the three parts it is made up of. Apart from flagella, there are two more structures. The first one is pili or pili, and then there is fimbriae. Okay, these are also surface structures, but they do not play a role in motility. Flagella, wherever present, it is help in providing motility or mobility, but the pili, cilia, fimbriae, these are not responsible for motility or mobility. Now, what is the pili? The pili or the pili are elongated tubular structures made up of a special protein which is known as pilin. Okay, the fimbriae are small bristle-like fibers that are sprouting out of the cell. In some bacteria, they are known to help attach the bacteria to the rocks in the streams and also to the host tissues. So the fimbriae, what is the function of the fimbriae? So the fimbriae, they helps the bacteria to attach to the substratum. Now that substratum could be a rock or it could be the host, okay? The tissue of the host. Now, is it clear up till now to everybody?
Yes, ma'am.
Yes, ma'am.
Okay, good. Now, talking about the ribosomes and the inclusion bodies. Okay. Now, in prokaryotes, the ribosomes are associated with the plasma membrane of the cell. Okay. And they are about 15 nanometer by 20 nanometer in size. And they are made up of two subunits, meaning 50S and 30S units. Okay. So, when these units are present together, they form a 70S prokaryotic ribosome. So the ribosome of the prokaryotes is 70S. Okay, and this S is the sedimentation coefficient. Okay, now the ribosomes are the prime site of protein synthesis. What is the function of ribosome? So it is protein synthesis. And then what happens, sometimes several ribosomes, they attach to a single mRNA and it forms a chain. For example, if this is an mRNA, many ribosomes will attach on it, okay? And it will form a chain. And this chain will be known as polyribosomes or polysome, okay? The ribosomes of a polysome, meaning the ribosome which is present in this whole set, they are responsible for the translation of mRNA into protein. What do the ribosomes do? They translate the mRNA into the proteins. Now talking about the inclusion body. See, the reserve material in the prokaryotic cells, they are stored in the cytoplasm because there are no any spaces, like no compartments, nothing is there. So these are stored in the inclusion body. The reserve food material, though they are present in the cytoplasm only, but they are present in the form of inclusion bodies. And obviously they are not bound by any membrane. Okay, since it is a prokaryotic cell, no membrane is there. And these inclusion bodies, they lie freely in the cytoplasm. What are the examples of inclusion bodies? So phosphate granules, Synophician granules and glycogen granules are such examples. Now, gas vacuoles are found in the blue, green, and purple and green photosynthetic bacteria. Okay, gas vacuoles are there. Okay, now this was about the inclusion bodies and the ribosomes. Okay, now let us talk about the eukaryotic cells. So the eukaryotic cells, the eukaryotes, it includes all the protistants, the plants, animals, and even the fungi. Okay. Now, in the case of eukaryotic cells, there is, there are certain organelles, there are certain, compartmentalization, meaning there is a division of the cell and each division has its own function to perform. Okay. So, in the case of eukaryotic cell, there is an extensive compartmentalization of the cytoplasm through the presence of membrane-bound organelles. See, now since every organelle present in the eukaryotic cell, they are bounded by a membrane. So they are present separately, even though they are present in like at one location, but they are present separately. That is why the word compartmentalization has been used. Now, the eukaryotic cells, they possess an organized nucleus, okay? An organized nucleus is there with a nuclear envelope. Now, apart from that, they have a complex locomotory and cytoskeletal structure present. Their genetic material is organized into chromosomes. And these chromosomes are the carrier of what? The genetic material. And what is the genetic material? It is the DNA. Now, all eukaryotic cells, they are not identical. The plant and animal cells are very different from each other. How? That the plant cell contains the cell wall, it contains the plastid, like the chloroplast is a plastid only. Okay, so it contains different kinds of plastids and a large central vacuole, which are absent in animals, meaning the animals will not contain the cell wall, it does not have the plastid, and it also does not have a large central vacuole. Okay, but in the case of animals, centrioles are present, which are completely absent in the case of almost all of the plants. Okay, now this one, you can see the diagram. This particular, the first one is how the cell looks like of an animal. Okay, and then this is, just a second, this second one would be the animal cell. Okay, why? Because in the case of the first one, you can find out that there are different sorts of chloroplast present here, as you can see. And since there are chloroplasts present, obviously this would be an animal cell, but here chloroplast would be absent. So this is an like animal cell, and wherever chloroplasts are there, that would be the plant cell. Okay, now moving forward, let us talk about the cell membrane of the eukaryotic cell. Okay, so the detailed structure of the membrane was studied only after the advent of the electron microscope in the year 1950s. Now, meanwhile, chemical studies were already being done on the cell membrane, and especially in human red blood cells. Okay, and this enabled the scientists to deduce the possible structure of the plasma membrane, like how it would be. Okay, they, it was, a sort of like approximation or estimation, how would the structure of plasma membrane look like at that time. Okay, so it shows that the cell membrane is composed of lipids which are arranged in a bilayer. Also, the lipids are arranged within the membrane with the polar head towards the outside and the hydrophobic tails towards the inner part. Okay, the polar head is towards the outer side, whereas the hydrophobic tail is towards the inner part, because it is needed to be protected from the water because it is hydrophobic. Okay, now this would ensure that the non-polar part, okay, the tail of the saturated hydrocarbon is protected from the aqueous environment, and the lipid component of the membrane mainly consists of the phosphoglyceride. It, as I said, that the cell membrane is composed of a lipid, so the lipid membrane, the lipid component would be the phosphoglycerides. Okay, then after, when there were like biochemical investigations done, so finally we got to know that there are also proteins and carbohydrates that are present in the plasma, like plasma membrane or the cell membrane. Okay, so the ratio of protein and lipid varies considerably in different cell types. Okay, the ratio of the protein and lipid varies. It is there. Both protein and lipids are there in the cell membrane, but the ratio varies. In human beings, the membrane of the erythrocyte means the RBC, which is the red blood cell. So the membrane of the red blood cell, it approximately has 52% protein and 40% lipids. Okay, 52% proteins and 40% lipids. Now, depending upon the ease of extraction, membrane proteins can be classified as integral or peripheral. Peripheral, as the name suggests, lying at the periphery, lying at the surface. So peripheral proteins lie on the surface of the membrane, whereas the integral proteins are partially or totally buried in the membrane. So depending on the ease of extraction. So which of the two proteins are easier to extract? Is the peripheral protein is easier to extract or the integral protein? Answer. Which protein is easier to extract?
Peripheral protein, because
Very good.
they're not
Yeah.
buried.
Yes, correct. Why? Because it is present at the surface area. It is present on the peripheral area. Okay, that is why it is easier to extract. Now, after all of these, estimations and everything, there was a model which was, very widely accepted. And this was the fluid mosaic model, which was given by two scientists. Singer and Nicholson in the year 1972. Now, this is a very famous model. Okay. According to this, there is the quasi-fluid, which is the semi-fluid nature of the lipid. Now, this enables the lateral movement of the proteins within the overall bilayers. So, since protein bilayer, like the lipid bilayer is there, and proteins move in between them. Okay, and this ability to move within the membrane. Okay, since things can move within the membrane, this ability is known as fluidity. Okay, so the fluid nature of the membrane, it is also important from the point of view of functions like cell growth, formation of intercellular junctions, secretion, endocytosis, and cell division. Now see, if a membrane will be fluid in nature, meaning it will be more flexible and it will, allow things to move through. So then only this properties like cell growth, then only it can expand, then only it can secrete. But if the membrane would be stiff, it cannot perform all these functions. Okay. Now, one of the most important functions of the plasma membrane is the transportation of molecules across it. The membrane is selectively permeable to some molecules present on the either side of it. Okay. Many molecules can move briefly across the membrane without any requirement of energy. Okay. See, whenever any type of molecule can move across the membrane and it does not require any energy, it moves across very easily. So this type of movement is known as passive transport. Okay. It requires no energy. What kind of molecules can simply move along? So those that are neutral solutes, they move by simple diffusion along the concentration gradient, meaning from anywhere there is a higher concentration to the lower concentration. Water can also move from the higher concentration to the lower concentration, but if water is moving, then we will not call it diffusion, but we will call it osmosis. Okay, the process, the movement of water by diffusion is known as osmosis. We do not call it simple diffusion. Okay, now the polar molecules, they cannot pass through the non-polar lipid bilayer. So they would require energy. They would require a carrier protein of the membrane to facilitate their transport across the membrane. Okay. Now, a few ions or molecules are transported across the membrane against their concentration gradient. Against the concentration gradient means from the lower concentration to the higher. So that type of thing would require energy. So that is why this process is known as active transport, a process which requires energy. And energy meaning ATP, since ATP is the energy currency of the cell. Okay, it is the energy currency of the cell. And what is the example of an active transport? So sodium potassium pump. As I told you, these are polar molecules. A pole is there, isn't it? But for the neutral solutes or for water, there is passive transport, then there is osmosis and simple diffusion. Is it clear to everybody up till now?
Yes, ma'am.
Yes,
No.
ma'am.
Yes, ma'am.
Okay, very good. Now we'll talk about the cell wall. Now, cell wall, it is a eukaryotic feature also, but in the case of plants, okay, not in the case of animals. So it is a non-living rigid structure, okay, and it is an outer covering for the plasma membrane in the case of fungi and plants. The cell wall, it gives shape as we studied earlier, and it also protects the cell from the damage and infection. Okay, it helps the cell in cell-to-cell interaction and provides barrier to undesirable macromolecules. Okay, now see, the cell walls, even it is present in fungi and it is present in the plants also, but their composition is very different in each of these. So since we are talking about now the algae, okay, algae is a type of plant only. So they have a cell wall which is made up of cellulose, galactans, mannans, and minerals like calcium carbonate. Okay, while in the case of other plants, it would contain cellulose, hemicellulose, pectins, and proteins. Okay, does anybody know what is the cell wall of fungi made up of? What is the cell wall of fungi made up of? Can anybody answer? Anyone? Siddhika, Martin, Alia, can anybody answer me? What is the cell wall of fungi made up of? Quickly. Does anybody know? If even if you don't know, you can say, ma'am, we don't know.
I don't know.
I don't know, ma'am.
Okay, it's all right. See, the cell wall of the fungi is made up of chitin. Okay. Also, the insects, you must know that the insect cell, that is the exoskeleton. But most importantly, the fungi, their cell wall is made up of chitin, chitinous cell wall. Is it clear? Now, the cell wall of a young plant cell, which is known as the primary cell wall. Okay. Cell wall of a young plant cell is known as what? A primary cell wall. Okay. It is capable of growth. Okay. It is capable of growth and which gradually diminishes as the cell matures. And then after the primary wall, there is a secondary wall formation which forms on the inner side. Okay. Towards the membrane side of the cell. Okay, so first primary cell wall is formed, then it gradually diminishes, meaning it ends, and then as the cell matures, a secondary wall is formed and it is formed towards the inner side of the cell. Then middle lamella is a layer composed of calcium pectate, which holds or glues the different neighboring cells together. The cell wall and the middle lamella may be transversed by plasmodesmata, which connects the cytoplasm of the neighboring cell. Okay, middle lamella is a sort of like a cementing material. Okay, and it is made up of calcium pectate. Okay, it glues the neighboring cells together. And then there may be plasmodesmata present in between so that there would be connection between the cytoplasm of the neighboring cells. Is it clear to everybody?
Yes,
So
ma'am.
for today's class, let's leave it till here only because I have another class now. Okay, so then I'll start from the endomembrane system. Okay.
Okay, ma'am.
Okay, ma'am.
Okay, then you people can leave.
Thank you,
Thank
ma'am.
you.
Okay.
Goodbye.